Good morning! Welcome to SR Life Science. Subscribe my channel. Let me know your opinion in comment box. And share with friends.
Plasma membrane forms the boundary of the cytoplasm, being guarded from outside by extracellular matrix. The membrane is responsible for the relationship of a cell with its surrounding. It is semi-permeable in nature. The detailed structure of cell membrane was realized only after understanding the chemical composition and the invention of electron microscope. A widely accepted model for the organization of plasma membrane was proposed by Singer and Nicholson in 1972 as the fluid Mosaic model. The model suggests a plasma membrane to be lipid bilayer surrounding the cell with a mosaic of globular proteins. The composition of lipid and protein varies in different cells. For example, human RBC contains approximately 52% protein and 40% lipids. Lipid bilayer makes the cell boundary in a quasi-fluid state, and it is dynamic in nature. Due to the fluid nature, lipids and proteins can freely diffuse laterally across the membrane. Lipid bilipids is composed of hydrophilic head facing the exterior and long hydrophobic tail of hydrocarbon chains occupying the interior of a lipid bilayer. Two different types of proteins have been identified in the plasma membrane, based on their location and association, i.e., peripheral and integral membrane proteins.
Welcome to the cellular garden! Peripheral proteins are involved in cell signaling and are Integral proteins are partially or fully buried in the plasma membrane. Transmembrane proteins are the most abundant type of integral membrane protein and are structurally, prokaryotic cell membrane is similar to that of eukaryotes. A special membranous structure is formed by extension of plasma membrane in the cell called mesosome, which is in the form of vesicle, tubules, and lamellae. Phospholipids form a bilayer. Non-polar regions face the core, and their polar head groups face outward, interacting with the aqueous phase on either side. Proteins are embedded in bilayer sheet, held by hydrophobic interactions between the membrane lipids and hydrophobic domains in the proteins. Micelles are spherical structures. These are aggregated in the interior, and their hydrophilic head groups are at the surface.
Check this out! The relative polarity of each amino acid is determined by free energy change. Wow! Accompanying the movement? Yes! This free energy can... Check this out! Membranes are composed of lipids and proteins in varying combinations particular to each species. Species? Check this out! Peripheral membrane proteins are loosely associated. They interact through electrostatic forces.
Like hydrogen bonds or lipid anchors?
Exactly! Covalently attached!
The quasi-fluid nature of membranes is useful for different cellular functions,
Such as cell division, cell growth, communication at intercellular junctions.
cell secretion, and endocytosis. The plasma membrane is selectively permeable,
It restricts molecular movement and maintains at intercellular junctions.
cell composition.
Integral proteins associate firmly with membranes by hydrophobic interactions between the lipid bilayer and their nonpolar amino acid side chains. Many membrane proteins span the lipid bilayer several times, with hydrophobic sequences of amino acid residues, forming transmarin alpha helices. The rate of fluorescence recovery after photobleaching, or FRAP, is a measure of the rate of lateral diffusion of fusion of the lipids. Flip-flop diffusion between brain leaflets is specifically catalyzed by enzymes called flipases, flopases, or scramblases. Wow, that makes a lot of sense! Integrane proteins, they attach cells to each other and carry messages between the extracellular and the cytoplasm. Specific proteins containing bar domains cause local membrane curvature and mediate the fusion of two membranes, such as endocytosis, exocytosis, and viral invasion. Specific prolin is an integral membrane protein that associates with the inner leaflet of two membranes, such as membrane transport and signaling. Invasion.
Some of the molecules move passively,
Without any expenditure of energy across the membrane along the concentration gradient.
called passive transport. Passive movement of molecules occurs by the process of diffusion and osmosis.
However, a few molecules either charged or uncharged cannot cross membrane by simple diffusion.
Movement of such molecules is facilitated by carrier proteins and channel proteins.
Such molecular movement is known as
The facilitated movement. Aquaporins are one of the critical channel proteins.
for transport of water in plant and animal cells across the plasma membrane. The protein composition of membranes widely, even more widely than their lipid composition, reflecting functional specialization. ABC transporters carry substrates, including many drugs, out of cells, using ATP as an energy source. Wow, that is fascinating! Ionophores are lipid-soluble molecules. They bind specific ions and carry them passively across membranes. That makes a lot of sense! Lipids and proteins can diffuse laterally within the plane of the membrane, but this mobility is by interactions with cytoskeletal structures. Wow, that's fascinating! Lipids in a biological membrane can exist. They exist in liquid-ordered or disordered states. Fluidity is affected by temperature and composition. That is good to know! Water moves across membranes through aquaporins. Some are regulated, right? Yes, and some transport glycerol or urea.
Some molecules cross against the gradient. This requires energy from ATP, just like the sodium-potassium pump.
This is active transport.
However, some active transports are ATP independent. Molecules are transported against the concentration gradients, with no energy utilization from ATP hydrolysis.
I see.
It couples transport of such molecule with a second molecule transported along. For example, active transport of ions
Sugars.
and amino acids.
Yes, sugars and amino acids.
In coupled transport, if two molecules are transported in the same direction,
Like the uptake of glucose and sodium, it is called symport.
if active transport involves transport of two,
Like transport of sodium and calcium by sodium-calcium antiporter, it is called antiport.
while facilitated diffusion transports only single molecules,
For example, glucose, it is known as untiport. The lipids and proteins of membranes are inserted into the bilayer, and they are inserted with specific sidedness, so they are functionally asymmetric? Multi-stranded beta barrels are also common in integral proteins in bacterial membranes. What about tire-dos of transmembrane proteins are commonly found at the lipid-water interface? One class of lipid rafts consists of sphingolipids and cholesterol. They have membrane proteins that are GPI-linked. That is a very specific subset. The myelin sheath consists primarily of lipids, whereas the plasma membranes of bacteria, mitochondria, and chloroplasts contain more protein than lipids. Cell adhesion, endocytosis, and the membrane fusion illustrate the dynamic roles of membrane proteins. Thank you for watching. See you in the next one. Stay tuned.